Okay, good afternoon, everyone, and welcome to our next session, the afternoon session on applications to neurological and mental diseases. And our first speaker is Jarek, who will introduce his own talk. Thank you.
Testing. Okay, working. Thank you, John, for the short introduction. It gives me more time to talk about actually what I'm going to talk about. And first of all, thank you, John and Hong too, for the organizing and the organizing committee and for the invitation. And what I thought I will do today, some of you might have seen portions of this talk, but I was trying to put everything kind of together for today. And for 15 minutes, it's sometimes hard to choose, what to put in there. So hopefully all the elements that need to be there are there. So it's maybe a grandiose title, Statistical Regularization used to study structure, function, connectivity, and everything else in the brain. But let me tell you what we've been doing since myself and my group and some collaborators over the past 10 years or so. So I won't really go over the outline, but basically what I will talk about today, kind of the past, is develop this peer family of methods. What's peer? Peer, it's partially empirical eigenvectors for regression. So we started that research working with imaging data, but not the imaging data that I will be talking about today. We started with magnetic resonance spectroscopy. It's anybody here in the room has done any work on MRS? Okay, so I won't go into that, but basically, we're trying to get signatures of metabolites, and this was in the brain. So we started that from the perspective of functional data. So in a sense, we have a signal, and then we have some external information, which are the characteristic spectra of different metabolites. So what we try to do, we try to use it in regression model. And then this was a few years back now. But what I'm doing these days is and here this is not exclusive. And I guess after the meeting or after the day yesterday, I know I should do all this stuff myself, according to Hong too, to put you on the spot, all the processing. I still rely on my collaborators who I kind of trust, I discuss it with them what they do. But again, I don't have neither the brain, no pun intended, neither the time to do everything kind of myself or kind of within my group. So, for example, for anatomy, we use usually FreeSurfer now with the parcellations going into hundreds, 200, 300, up to 1,000. I mean, hopefully FreeSurfer will go there, but so far we're using the cortical parcellation of the Destrue parcellation 148 parcels. With structural connectivity, again, I have a good collaborator who I will mention at the end. So he's processing this data. It's mostly using kind of diffusion tensor imaging methods, but also going further into more modern methods of processing such data. Functional connectivity, we're still relying on correlation, but can use other ways as well. And then with Cortex, we are also looking at the anatomy. So this is actually I like this picture, actually. This colleague who I work with created this picture, so I borrowed it from him. Okay, this one. So basically, you're going to the scanner, and then you can get a lot of different. So I'm working with MRI data. You can get a lot of different information that then can be kind of mapped to either connectivity matrices, either functional or structural, or the kind of cortical properties. But now, what do we do with such data? Well, it's all extraction of the information and so on. So we talked already a lot about that. We extract connectivity. But what are the questions that we're actually trying to answer? And going with the theme of the session is kind of, can we actually gain some information from the brain that will help us either with associating that with the cognitive outcomes, or maybe predicting future outcomes. But the most important part that we're doing in our work is kind of we're trying to do this, shouldn't call it multimodal, but borrowing information from one kind of mode. Let's say, can we borrow information from structural connectivity to tell us something about functional? Or can we borrow something, from one of them for the anatomy to help us in associating, let's say, structural connectivity with the outcomes? So now, a lot of work, and this is, again, a bit grandiose. We've done a lot of work on nodes or ROIs. Yes, then with connectivity, we move to edges. But there are also higher order structures, and I see Mo standing there. I'm calling people here who worked a lot with higher order structures in the brain. So, for example, with triangles or tetrahedra or pentachora or the other structures going into higher dimensions. So the question is there information there? Does the information lie there? So what we do, we start with a very simple model where we're trying to predict an outcome from the information that we have, let's say, from anatomy, but using this additional information from here, for example, structural connectivity. Now, what's nice about that? Well, we can incorporate that in a penalized or regularized regression framework. So usually how we would solve such a problem is, in a kind of simple continuous outcome, we would add some penalty to our least squares criterion. And there have been tons of penalties proposed, starting with rich estimate and lasso and kind of generalized versions of those. But this paper that I mentioned at the beginning, we use this structure penalties for functional linear models, partially empirical eigenvectors for regression. So we use MRS as an example in that paper. So basically what we want to do is we want to use information, this external information. So for example, here through Laplacian of the connectivity matrix to inform the estimation of the coefficients or the associations that we're interested in. Now, the nice thing here is for L2 type penalties, we can make this equivalence with the mixed models. What does that give us? Well, one of the things that's always hard in the regularized regression problems is the estimation of the tuning parameter, lambda here, okay? Now, with this equivalence, we get for free the estimation of this tuning parameter because it's just the ratio of the variance components. But also we can borrow some theory, computation, and basically methods that have been developed for linear mixed models over the past 40 years. So this was kind of I don't say the first proposal that we've made, because the first was really working on functional data, but in a sense, this ordinary rich regression would be just B transpose B. Yes, this would be just the sum of squares of the coefficients. But here we have those weights, and those weights are informed by external knowledge. Now the question is, does this external knowledge actually help us? We don't know. So the next version that we published was, and what I said, this peer family of methods. We have repair, now we have gripper, we have despair. This was the last method, didn't know what to do. We were despairing, so we proposed it. So basically here, we do it in an adaptive way, where now we have two penalties. And here, if there is any information, well, we get this information. If there's no information, we'll just revert to the rich penalty, okay? So in a sense, in a situation where there's no information, we penalize everything kind of equally in all directions. But if there is information, let's say if the coefficients are close to each other, well, our penalty kind of surface is of oval here, so it prefers certain directions over other directions. So again, we can connect it with mixed models, and we extend it, as I said, to this despair. So here we use additional information of the distance between the cortical regions, where now we have three penalties. Now, we extended that also to no Gaussian distributions. So this was published a few years ago, but what I want to really talk about a bit more, kind of going beyond this was still information on the nodes. Yes, this was information on the cortex. We might have a property of the cortex, let's say cortical thickness. What if we want to actually do scalar on matrix regression? Well, here the estimation is, it's not of the regression coefficient vector, it's regression coefficient matrix, yes? So now, one of the things, and we had those, I don't say issues, but we had let's say when you write a paper, submit a paper, and people say, oh, you try to solve this problem in general. And that's not what our goal was. What our goal was, for specific situations, we can provide better solutions because, again, we might gain something from either external information or from the assumptions. So here, one of the assumptions, and this is, again, going with the kind of brain imaging literature, that things in the brain don't happen kind of randomly, let's say sparsely throughout the whole brain, if this is a connectivity matrix. But there is a high chance that they are kind of in blocks or in networks. And these networks don't necessarily, so these are like intra-network connections, but there could be inter-network connections. But again, we're looking for those structures. And one of the ways to get at that is, again, to use penalized regression, which works for both low rank estimation, which will give us actually this the structured solution, but also we want sparsity. So I won't really present any results today. I have a list of papers at the end that you can look up. But basically, this is here, we're not using additional information, we're using additional assumptions to get at the solutions or to get close to the solutions that we want to get to. So, sorry. So for this kind of the peer-based methods, it's, we are, again, trying to use this a priori information, but we're doing that in a principled way, so through the penalties. Now, the nice thing there is these penalty parameters we can choose in an adaptive way via maximum likelihood methods. And here, as I shown briefly, we use these different sources of information. So let's say structural connectivity, functional connectivity, distances, and so on. Now, this is kind of addressing one of the questions, at least, that was posed in the email, kind of future directions. So one of the things that we haven't done yet, that this is something we're working on currently, is we introduce certain assumptions for the scalar on matrix regression, but we haven't incorporated this external information. So this is something that we are pursuing currently. And then this thing of this higher order structures, this is just something that, I mean, we started working maybe 2-3 years ago on that. It's a hard topic, so just taking us some time. But we know nodes are important, we know edges are important, but triangles, this is kind of this next, it's seemingly simple step, but it's working with the kind of this k-order simplicial complexes. Again, it's a bit more complicated. So this is one of them, is kind of detection of these higher order structures. It's not only, stopping at the edges. And then with the penalization, so going to the first point, it's using kind of this hot Laplacian matrices in regularization. The one thing that, something I talked to a few of you here about that, and I hope to talk to you more later about it, is how to deal with directionality where we don't have directionality really in the connectivity matrices. And of course, it would be great to get some inference and interpretation. This is more like scientific community versus inference. That's, I guess, more our community. So people who I work with, some postdoc students and collaborators, my two main collaborators on this project are Tim Randall from Fred Hutchinson Cancer Institute and Joaquin Goni from Purdue University. And there are other, as I said, students, former and current postdocs, and just some literature. So thank you.
Thank you, Jarek. I think we have a time for a question, if anyone has one.
How do you validate the findings in fact?
So one of the things we did, so two levels of validation. One of them is kind of more of a computational statistical validation via resampling, and this is internal validation. But then, of course, testing the stuff, well, we can always do simulations, yes? So if you assume something, if you recover it, great. But the testing the method or applying the method to different diseases, different, outcomes, that's just something, it's at the end of the day, as I said, there is this scientific interpretation of the findings. If you find something like in visual cortex that has to do with, I don't know, alcohol use, it's probably not the correct finding.
Deliver some imaging biomarker to other people?
I hope so. I hope so. We're actually, so we applied this method to alcohol use disorder data. And we found some, with the connectivity, we found some interconnectivity patterns that were not actually found earlier. So this is kind of like the next step to, look more, not over the whole brain, but over, specific regions or specific areas of the brain that, it's like for the next experiment. Thank you. So since John is the chair, this is John.
That's a good introduction.
Thank you. And thanks for the awesome introduction, Jarek. Much appreciated. So, title of my talk, disease progression modeling for frontotemporal dementia. I'm kind of cheating here when I say it's with all FTD and the Berry consultants because they really did all of the work and I just kind of gave opinions on stuff. So the credit's all theirs, and all of any criticisms you have also go to them as well. Oh, we're kind of stuck here. Okay. And I should thank, of course, Hong-Tu and Jan for all of their work in doing the organization. Even though I was on the organizing committee, I did nothing. They did everything, so they deserve all the credit. I have an apology to start with that even though my talk was about disease progression modeling, I'm going to spend quite a bit of time talking about the bulk response, which is completely unrelated, but it's prompted by Jan's questions and the discussions yesterday. And so I'm going to take advantage of talking about that. And anyway, Tanya in her talk this morning talked about disease progression models, so I don't need to so much anymore. So I'll talk about the bold response, tell you a little bit about frontotemporal dementia, what disease progression modeling is for us, the problems, difficulties we have, and then I'm going to, all the way along, I'm going to focus on Jan's questions. I'm kind of putting my answers up front here. And this is all my opinion. But what are the most important research problems we should focus on in the next 5 years? I think we need to better understand and model the bold effect in functional MRI. I think it's a really important problem that's been neglected. And completely different problem, how do we define disease onset for various kinds of dementia? Tanya had in her talk where she was basically anchoring things on a diagnosis time. Whatever you want to call it, there has to be that anchor there if we're going to understand these progressions. Overall, what are the important areas? Kind of goes to Rob's discussion yesterday. We have to interact with people in other scientific fields, and I mean, really interact with them to solve these problems. Actually, that's my answer to 3. So And then two, I'm going to leave to people like Hong Tu, who are much more knowledgeable about how to do dissemination of data sets. Okay. So when I talk about the bold response, these are my own opinions. I take full responsibility, and I really have no right to be talking about this at all because I've not worked with functional MRI for over 20 years. But I think some of these opinions still hold. Yeah. So fMRI, I'm sure most of you know, has been around for around 35 years, back to the original papers of Ogawa. And despite all that time having passed, the bold response is still mostly modeled in these ways as an experimentally invariant model. It's the same shape, all conditions. There are some exceptions I know, but in practice, spatially invariant, we have the same model across space. It doesn't change no matter how large the response is, the shape stays the same. It's subject invariant, we're all assumed to have the same hemodynamic response shape. And it's modeled primarily by convolution, which is a surprisingly strong constraint. In addition, we model it voxel by voxel typically, so it's a localized response estimate. But really, it's a spatiotemporal process, the bold response, so that's obviously not a good model. So we could do better to understand this, but we've got to work directly with biologists, biophysicists, clinicians, etc., to not just to get data from them, but to work with them to design experiments so that we can statistically analyze that data and learn more about what the response is. So here's the problem with convolution. So standard model most people use, kind of in SPM and stuff. At least I think that's what they still use. It's sort of like virtually uncontested since the mid-90s when SPM came along. I mean, they needed a quick model to work with fMRI and kind of justified it by this match filter theorem that I spent a long time trying to figure out what the hell the point of that match filter theorem was. They seemed to use it as like, this means that the model is great. And I just couldn't understand it. I was a PhD student at the time, and I still don't understand it. So what's wrong with the convolution? So here's your sort of typical hemodynamic response function. And I should say, when I'm talking about biology, I'm not a biologist, so it's kind of pretty wishy-washy, so don't hold me to everything I say. But we have a the MR signal, you have a stimulus onset, you get supposedly an initial dip where this local oxygen is being used up. And then you get this big responsive blood flow that replaces the oxygen that's been used up and overcompensates, and then it drops off and it goes, you have this negative overshoot before it settles back down. So that's what people think the response looks like. But this is what it looks like when we put it in a convolution. It doesn't look anything like this. You have scans, let's say these are taken every couple of seconds. And then when the acquisition starts, you start adding in terms from the hemodynamic response. And then when the stimulus ends, you start subtracting them in the same way that you added them in the first place. So you have an anti-symmetry brought in, and this is not anything I discovered. It was first noted as far back as 1997 by Krellin, Johnston, and Hasey. Notice here, this shape does not vary by experimental condition, doesn't vary by stimulus length, doesn't vary by subject, response level, or spatial location. In particular, response level is kind of an interesting one, because if you have a voxel that has a very large response, that means it still has to shoot up with the same pattern, and then it has to drain away at that same rate, again with this anti-symmetry. And if you've ever filled up a sink, like to the top versus just a little bit, you know that the drainage time depends on how much stuff you put in the first place. There's more problems with the bulb response as we model it so far. Spatial displacement. I don't know if you've heard the phrase about watering the whole garden for the sake of one flower. People used to say this a lot in the 90s. So the idea is you have some of the pull out of your brain, activation occurs, some kind of biological signal, say send some blood flow up there, it goes up through your arteries, kind of like along a branching tree, gets to the capillary bed, replaces the oxygen, but it overcompensates and it spreads out around the region of activation, and then it decays away through the venous system. So this leads to non-local signal, obviously, because it's spreading out. It's not going to be isotropic around the activation necessarily. It's coming up through this arterial system. So kind of like you could be watering it from the side or from the front, and you'll get a different pattern. So that's why I use the word smear as the activation signal. And it's differentially delayed because if you're early on in that arterial system, you're going to get your signal, you're going to get your blood flow before somewhere later on. Depends how far does it have to travel to get to where it needs to go, and how fast is it traveling along that pathway. So it's a really complex response pattern. It depends on the arteriovenous system. And even though I'm talking about bold response models, this also, I think, matters for connectivity studies too, that people have been talking about a lot. So does it matter? I think I've already said, yeah, it does, and it does for sure. Basically, it reduces detection power, it increases false positives because of the spatial spread thing. You kind of lose an opportunity for understanding the brain because we're not learning about where the activation really is. It raises questions about connectivity studies, like a default net. I know people there was comments yesterday about not overstating what connectivity is, that it's just correlation, but I think that's a bit of a cop-out. We can't just forever say, all we're doing is measuring correlations. We really need to put the effort in to understand what exactly are we measuring and what's the point of it. So if you have this kind of complex branching system, you can have regions that activate at the same time or a little bit offset, and you might think they're talking to each other, but they're not at all. It's just coming along the same pathway. Okay, so back again, we need to work directly with physiologists, clinicians, blah, 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 to try to understand this very complex spatiotemporal signal. We will need to get people to kind of make these image acquisitions, which they can do in the MR scanner at the same time as your fMRI, and get these angiograms, venograms, kind of correlate pixels to determine relative delays in different positions. It's a lot of work, and I think it's been neglected. Okay, now talk a little bit about in 3 minutes, what I was originally going to talk about, frontotemporal dementia. So kind of another form of dementia you may not have heard of. It happens early in life, unfortunately, to people that get it. It's pretty rare, 10% about of relative to dementia. Affects younger people, 40s and 50s, mostly. It's also very heterogeneous compared to Alzheimer's. We have like behavioral variants where you start behaving inappropriately, language variants where you can't find the words you want to say, or you know what the object is, but you can't pick the word for it, or the other way around. You know the name of the object, you don't know what it's for. And then there's also motor presentations you might be familiar with, like ALS, PSP, and CBS. We're not quite in the Huntington's disease position where everybody has a genetic marker that they have the disease. We do have genes identified for about 30% of cases. So we call these familial FTDs, and they tend to be hereditary. There's also 70% are sporadic, not necessarily any family history. It's just sort of random appearance. But the familiar are obviously very useful potentially for studying progression and learning about if we want to get people into clinical trials early in the disease. This is a particularly useful set to try to understand. Okay. We're part of a large consortium, the all-FTD study, centers across the US. And then there's also a European version and a British version. And so there's some sort of like they're collaborating. We get a lot of cognitive tests, imaging, primarily structural MRI, but some other stuff too, blood work. Lots and lots of people involved. And let's skip this bit. So we have this joint model of endpoints. We have kind of a cognitive score, various neuropsych tests, neurofilament light, which is a blood biomarker, and then volumetric regions of interest. And here's the difficult part, the predictors. Really, it's based on disease age. But what does that even mean? How do we even define what a disease age is? What does an onset of disease mean? Even how do we get a working definition that we know is the same for every individual? That's the difficult part of this problem. We basically have some model to do this kind of curve stuff, but I'm going to skip right through that. Again, the tough part is the disease age there. And even though sort of clinicians can come up with an age of onset, there's a lot of variation there. I'm just going to say these models kind of fit. We can come up with parent sample sizes, but I don't trust them because of this disease age thing, the age of onset. The model gets driven a lot by kind of trajectories that we're not even really sure. We rely on the model with it too much, so I have a lot of concerns. That's exactly 0 seconds there, so I'm going to walk away and pass it on to the next speaker. So quickly switching back to chair mode here. Our next speaker is Dana Tudorascu, University of Pittsburgh. I'm actually going to say her title, The Statistical Challenges in Analysis of Tau PET Imaging Data in Alzheimer's Disease. Thank you.
Thank you, John. I would like to thank the organizers for inviting me and for organizing this wonderful conference. So I will talk about specifically the questions that we were asked to talk about in the context of PET imaging data, because that's what I've been doing in Alzheimer's disease for the past couple of years. And I will give you my perspective in terms of how can we make statistical methods more impactful and sort of like my own experience and how I sort of like penetrated the clinical world, because I go a lot to these clinical conferences. So to sort of understand where I'm coming from, I am an applied statistician. I work with a lot of multi-site studies. Okay, so one of them is the Alzheimer Biomarker Consortium. This is the Down syndrome. So we're studying Down syndrome in Alzheimer's disease. Actually, Alzheimer's disease in Down syndrome people. This is a multi-site studies, 8 centers in the United States and Cambridge, UK. And I direct the data core. I am also the biostatistician for the head-to-head harmonization of tau tracer in Alzheimer's disease. This is the head study. This is the first study in the world that is collecting multiple tracers in the same individuals. This has 6 US centers, Canada and Spain. I am also part of the ADME 4. I'm a co-investigator, but in a very small portion of the ADME, and that is developing a single universal scale for all tau tracers with the same goal as that of amyloid, which is the centiloid scale some of you might be familiar with. And the other study that I'm involved with, also a multi-center study, is the diverse DCID. That is a vascular study, and it has 19 US centers. Dr. DiCarli at UC Davis is one of the PIs, and I'm a co-investigator in that, specifically working on data harmonization. So PET imaging in Alzheimer's disease, some of the advancements and some of the challenges is that the reason we talk about all these harmonization is that we moved from one-site studies to the multi-site studies. So we collect images under different conditions on different machines. And then one of the major challenges that has been now surfacing in PET tau imaging is the fact that we're collecting images using multiple tracers. And what are the challenges with that? So we started talking about this. Martin talked about this, the software that we're using, the pre-processing, the platforms, the pipelines. And then in these multi-site studies, one of the things that we're sometimes facing is that we use the derivation of these imaging outcomes at multiple sites, and then we want to put them together. Then we use, different sites might use different data. That's true in some of our studies, and with different tracers, and then we want to combine them together. And in the PET imaging world, I take full responsibility of that. Smoothing is the actual standard of data harmonization, and I can tell you that it does not work. What is PET imaging? So PET imaging is kind of like the baby that I started with when I was a PhD student. So PET is a molecular and functional type of imaging. So you're collecting data based on injecting somebody with a radioactive tracer in their blood. So one thing is that you have to wait until the tracer reaches the blood and then enters into your brain, and then it's measured. So for example, here, during a certain period of time, so this is the posterior cingulate area, one of the areas that are important in Alzheimer's disease, but it has to be calibrated with a region that doesn't uptake, the tracer. So in this case, so we have to, because PET has a very low resolution, we have to use MR data to rely on to co-register with, to figure out the regions that we're interested in. But one thing that is of major concern these days is the fact that, for example, tau, which is a protein in the brain that is found to be responsible for, disease, for the Alzheimer's disease, is that could be measured with different tracers. The issue is that the tracer distribution is different in the brain. So although they are measuring the same thing, this is the same person that is measured with fluorotaucipir tau, which has been approved for the FDA to use in Alzheimer's disease clinical trials. And then there is the MK6240, okay? The same brain, as you can see, very different image. So I was just telling you a little bit about this. So tau proteins are present in the brain of people with AD. And these tau areas of accumulation are actually categorized into the Braak stages. The 1 and 3 are sort of like early accumulation stages, and 4, 5, 6, and they are later accumulation stages. So the tau quantification and PET imaging, going back to the pre-processing. So there is some sort of a standardized way of measuring this accumulation, okay? So the SUV, or standardized uptake value, is measured by the average concentration in some specified, volumes, some area of the brain, and is measured in kilobecquerel per milliliter, and then is divided by the injected dose and the body weight. And then there is something that is called standardized uptake value ratio, where you take this SUV and you divide by the cerebellum, because the cerebellum in tau imaging, it does not accumulate. So it's free of the tau. Some of the challenges, as I was saying earlier, is the off-target binding. So tau accumulates in the choroid plexus, right? In this area. This is, again, this is the same subject that is measured with the FTP and MK, so this data for the head study. And as I said, we haven't done this before. So you can see there is the distribution of the tracer is not the same between FTP and between MK. Striatum area and meninges. Now, the problem is not only that, but these areas are not related to the AD pathology. They are actually characteristics that vary based on individuals, and they are artificially increasing or decreasing these SUVRs that we use as outcome measures to make inference statistics, okay? There are some harmonization methods for the tracers, and one of them is called Centaur-Z, which started in about 2023. They work in some instances, and some instances they do not work. So what should we focus on in PET imaging in Alzheimer's disease and why? And again, this is my opinion and the struggles that I've been dealing with for recent years is that, you know, this multi-site integration of the studies, okay, and PET imaging tracer standardization. One of the things we've been talking about is that should we even combine them? I mean, the distributions is different. And you know, I would like opinion of people that know a lot more in terms of chemistry and physics about this. And I work with a lot of people, and there is a lot of discussion if we should we even combine them, and thinking about clinical trials, right? Should we scan people at one site with one tracer, at another site with a different tracer? I don't know, maybe we shouldn't, but I think that's where we're going because of these push for these huge multi-site studies. And then a gold standard kind of like for PET tau imaging doesn't exist. Now there is a traveling Hoffman brain phantom that has been used for ANI for PET MRI study calibration. It doesn't actually work for PET, okay? So it does a pretty good job for MRI, but for PET imaging, it doesn't work because the phantom has a single chamber and it represents the whole brain and cannot account for off-target binding that I've just shown you. And cerebellum is part of that phantom. So we just have a single compartment, right? Also, recent studies have shown that the Hoffman phantom works for FTG PET. FTG PET does not have a single region for calibration, and the SUV quantity can be used in analysis. And so far, I do not know of any existing methods for integrating human PET imaging replication studies with this phantom data to kind of figure out where are we and what is the actual truth. What have we learned from the HEAD studies? And now the HEAD study has this, the goal is to accurately quantify these tracers so that they can be used in clinical trials to measure treatment effects while being cost effective. So I'm going to show you about 180 participants, cognitively unimpaired, mild cognitively impaired, and ADs that were scanned with 2 tau PET tracers, FTP and MK, 6 sites, and 6 different scanners. And I'm going to show you some of the, effect sizes that I have computed. Cohen's D for FTP and MK on smoothed images and just using combat. As you can see, so here is the MK, CU versus MCI, smoothed and combat. These are the effect sizes, in parentheses, the sample sizes. One thing that I want you to pay attention from a clinical trials perspective is depending on the method that we are using, you are looking at costs between $16,000 and $112,000 difference based on the method, okay? So this is PET scanning is very expensive, so that can actually make a difference. Now here, those effect sizes, they are large, but think about smaller effect sizes and how much difference that's gonna make. For the FTP, of course, between smoothed and combat, of course, the effect sizes are varying, sample sizes are varying, the difference is in cost between $8,000 and $48,000. And now between tracers differences between no difference and about $64,000. And that's just based on the method that you're using, smoothing, combat, whatever it is, that variation that you would like, is just that being non-consistent, you can see these differences. So how can we make statistical methods more impactful? Again, this is my perspective. One thing that I've learned is that participating in clinical meetings and giving presentations is very efficient. Presenters at a statistical methods conference with clinical focus, right? From an Alzheimer's disease perspective, AAIC, human amyloid imaging, clinical trials in Alzheimer's disease, AD, Parkinson's disease, satellite AAIC symposium. You write an abstract with a clinical focus, and then you go and you present a method, you relate it to the clinical data, and use interpretation and inference, and you can talk with the clinicians in their own language. Ask your collaborators to be there and support you and call them out when you are asked questions, in the clinical world. And they tend to have them in really nice locations. And then I think one thing I learned is that at your institution, it's good to participate in organizations that are clinical focus specific, okay? We have ADRCs, behavioral health centers, sleep centers, and they have these related NIH agencies and organize meetings with NIH officials. So to participate in their annual meetings, to have posters, to talk to the NIH representatives and tell them about your work, why is it important, and they do actually listen, show the importance of the methods, speak with them in terms of clinical effect sizes, clinical significance, clinical trials, importance of the cost reduction by using the correct methods, and write a grant proposal with your collaborators, add their clinical expertise. That's always helpful. So I think, in my opinion, PET imaging needs more AD, and AD needs statisticians, participation in clinical conferences. It's, important. And how do we know when we are making a successful impact? I think when our clinical collaborators use the methods we suggest or adapt them, or the methods that we have developed, and when they ask us to be involved, not only in the year 4 or 5 study, I've been asked year 4 and 5 to just analyze their data, but from the beginning, after they started collecting their data and from planning stages to the end, every single step of the way. So these are my collaborators, and thank you. And one thing I think, one of the major contributions, and this is what Jarek told me, and I agree with him, I think combat method has made a really huge impact in the neuroimaging world. Every single grant has that, combat harmonization. So that is where the statisticians have played huge role, Taki's group and Johns Hopkins. So thank you.
Thank you, Dana. We do have time for a question or two. That's it. On to, go ahead.
Yeah, it's great. But in the tables, you show the comparison between combat and how do you know that combat method is really working in one sense?
That's what we don't know. So because as I was saying before, we're kind of missing what a gold standard is, and some of the studies that we've been doing, we do not know exactly the variation that is coming from scanner differences, from pre-processing pipeline. That was just an example by using, smoothing and combat. But combat, the way people have said that it was successful, it's actually increasing your effect sizes, and it's decreasing the variability across.
It's possible to develop some data set?
Yes, we are working on a multi-chamber phantom, PET phantom that we are planning to scan at the same site, some of the sites with ADNI and some of the sites with the head data where we have paired, tracers. And that phantom has, you can inject it with multiple different tau tracers, as well as with, you can account for the cerebellum.
Okay, thank you.
Thank you.
So final speaker of the session is Duygu Tosun, and she will talk about multidimensional biomarker landscape in Alzheimer's disease and give insights for improved disease modeling and clinical trial design. Thank you, Duygu.
Thanks, John, for the introduction. I'm going to have a slightly different style presentation from more clinical perspective, some of the challenges and how methods can be applied in clinical trials and clinical practice. So the concept of dementia is very old. It dates back to the civilization. And currently, and even back then, the way we diagnose dementia, Alzheimer's disease, really is based on cognitive measurements and the clinical assessments. But through these years, with advancements in technology, starting with Alois Alzheimer and his colleagues observing that amyloid plaques and tau tangles, allowed us to have this biological definition of the disease, which suggests that it's what we call an amyloid facilitated tauopathy, meaning we observed amyloid accumulation first. That leads to downstream tau pathology, accumulation in the brain, which leads to neurodegeneration and which leads to the cognitive impairment and the clinical symptoms and the clinical diagnosis. It's a very simplified biological model, actually, that there are lots of unknowns. But what these kind of biological observations and the definition of the disease allowed us to is this shift from syndromic definition of the disease to more biological, biomarker-based definition of the disease, which actually tackles some of the challenges mentioned earlier today about how we define a timeline, how we align the observations in a unified time scale. But also that changed the clinical trial landscape. Rather than recruiting participants based on their symptoms, when we look at the current ongoing and most recent clinical trials, they started to incorporate biological definition of the disease in their clinical trials. And this is really important because the trials actually target a particular biology. They try to remove, for instance, amyloid pathology or tau pathology. And they do really need to have those pathology present in their participants to show the efficacy of their treatments. So this is a snapshot, which is relatively old at this point, but pretty much all of them screen the participants based on their clinical measures and cognitive measures. But in addition to that, they also want to justify, validate that the participants actually have the AD etiology, the true biology of the Alzheimer's disease. So when we look at the current landscape of the biomarkers, you've been hearing the amyloid, that is our first hallmark pathology. They need to have that to be in that AD spectrum. We also have tau biomarkers for the tau pathology, which could be based on the tau PET imaging or some of the fluid biomarkers we currently have. And then we get into the biomarkers that looks into the neurodegeneration, which is more related to their symptoms, their cognitive decline and clinical symptoms. So it's this ATN framework we keep talking about in the Alzheimer's domain. So one of the things we did actually with this establishment of the biological definition of the disease a couple of years ago is we looked at to what extent these key hallmark pathologies, the biomarkers we have for those pathologies actually explain the variance we see in their cognitive decline. And we use lots of imaging data, amyloid tau PET imaging, and then we also use MR imaging for neurodegeneration, and also we use FLAIR MR imaging for potentially comorbid cerebrovascular pathology in the brain. And not surprisingly, what we observed was we can only explain a certain percentage of the variance in their cognitive decline with all these, what we call the ATN biomarkers specific to Alzheimer's disease. And again, this wasn't a surprise because there's this huge literature, autopsy literature, repeatedly reporting this. But this was the study, one of the first studies where we actually use in vivo biomarkers in living individuals to re-show that these ATN biomarkers, they only explain a certain aspect of the disease, not the entire story. At the same time, it also re-emphasized the heterogeneity we are seeing in this disease. Yes, it's true, they need to have amyloid to be in that AD spectrum and to be called, they have the AD etiology, but everyone is presenting and experiencing the disease differently. And one of the things, again, coming from autopsy studies is some of the variants we see in their cognition and clinical symptoms are driven by their comorbid conditions, especially the other co-pathologies they have in the brain. And again, this is, there's a huge literature coming from autopsy studies showing that it's actually very rare to see an Alzheimer's disease brain just presenting with amyloid and the tau pathologies. Pretty much everyone presents with some additional co-pathologies in their brain when we look at this at autopsy. And some of them are more common than others, but it's a very long list, including cerebrovascular pathologies, Lewy body pathology, TDP-43, CAA, and it goes in and on. And again, it's the notion. And the important thing is, again, coming from autopsy studies, all of them actually contribute to their cognitive profile. So this is one example study where they look at the global cognitive function trajectories before death and try to explain which pathologies actually contribute to their trajectories. It's true that AD pathology is the major one, But the other pathologies, especially the Lewy body pathology, are actually contributing significantly to their cognitive impairment, introducing more heterogeneity. And it was replicated by many, many other autopsy studies. And but the problem, the challenge here is we have all these biomarkers for the AD pathology, amyloid tau, and to some extent neurodegeneration. But for some of these co-pathologies, we don't actually have biomarkers, especially for Lewy body, TDP-43 and CAA. Those are the key ones that are clinically relevant because they do contribute to their symptoms and their cognition. So another study we recently did was leveraging autopsy studies. Some of these autopsy studies, they actually have antemortem imaging done on the same participant. So we put together a combination of multi-cohorts where they have neuropathology assessments at autopsy and also have neuroimaging data collected antemortem, and use some machine learning approaches and also leverage the peer approach, actually, in this case, for the regularization. We built models to impute the presence of some of these co-pathologies for individuals that are still alive and participating in our studies. And one of the questions we asked, going back to the earlier result I showed, can we actually explain more of their variance, more of the variance in their cognitive decline and clinical decline? And what we observe with this, again, with the AD-related pathologies, we can explain a bigger portion of their variance in cognitive decline, but the co-pathologies, actually, the non-AD co-pathologies explain an additional 13 to 21% of the variance in their cognition. And one of the strongest co-pathology was the Lewy body pathology, which was consistent with previous autopsy studies. And most of my work is related, linked to clinical trials. So we're really interested in what does this mean in clinical trial settings. So if we know the pathological composition of each individual, meaning their pathology profile, do they have AD pathology, but if we also know if they have co-pathologies in their brain, will this allow us to design better, more efficient trials? And this was one of the simulations using this machine learning-based imputation for co-pathologies, showing that, yes, if we know and if we account for the contribution of co-pathologies to their cognition, we can actually have, we will need a smaller sample size to show the same efficacy for a given trial. Since then, if you're following the AD research, actually, there's lots happening in fluid biomarkers. This is the new thing for us beyond imaging. And one of the new advancements in fluid biomarkers is this new technology to detect the Lewy body pathology in fluid, in cerebrospinal fluid, CSF. And again, the Lewy body pathology is actually one of the frequent co-pathologies we see in Alzheimer's disease participants, patients at autopsy. It's what we call a seeding assay. It doesn't actually detect the amount of Lewy body pathology, but it detects the alpha-synuclein seeds, which are related to the Lewy body pathology, and it's currently a binary detection approach. It tells us if the participant has alpha-synuclein seeds or not. But although that is a limitation, it still has this huge clinical significance in terms of, for us to understand the role of Lewy body co-pathology in Alzheimer's disease from individual's perspective. And this is one of the recent studies where we showed with age, we see greater prevalence of Lewy body co-pathology in Alzheimer's patients. It is related to the disease stage. It is related to the cognitive impairment, and it's also related to the AD pathology. But those relationships are actually not straightforward linear relationships. When we start looking this in more detail, for instance, when we look at how these pathologies are changing with age, when we look at cognitively unimpaired or mild cognitively impaired individuals, there's that expected increase with age. As they get older, they show more AD biomarkers, biomarker burden, and as well as more alpha-synuclein Lewy body pathology. But when we look at the dementia cases, the story is more complicated. If they are younger with dementia diagnosis, they tend to be present with more pure AD pathology, meaning they have the amyloid and tau, but not necessarily Lewy body. If they're older, they might have lower amyloid and tau pathologies, but still significant, but they present with co-pathologies like the Lewy body pathology. So there's that complicated nonlinear landscape of how these pathologies are interacting. And at the same time, when we look at, from that perspective, how different pathologies are interacting, what we're observing is with greater amyloid burden in the brain, they are more likely to be Lewy body pathology positive, but the story is different when we look at their tau pathology. What we're seeing an inverse correlation. If they're SAA, if they're alpha-synuclein positive, they have lower tau pathology. Doesn't mean that they're protected against tau, but it really tells more about this mixed pathologies and the contribution of different pathologies to the symptoms and lowering the threshold for cognitive impairment. We've been looking at the timing of these pathologies, and this is a very special cohort. We have a small subset. They've been developing Lewy body pathology during their involvement in the ANIS study particularly. And we are using what we call the amyloid age. We are using amyloid as our timeline to look at if amyloid is happening before alpha-synuclein Lewy body pathology onset or vice versa. And what we're observing is it's really the amyloid that is presenting first before the onset of Lewy body pathology in cognitively impaired individuals, not necessarily in cognitively unimpaired individuals. And being amyloid positive is, and also being an ApoE4 carrier, homozygote carrier, are high risk factors for acquiring Lewy body pathology. And finally, the last thing I want to, from results perspective, just going back what this means in terms of clinical trials or understanding the heterogeneity we're observing in these individuals. Again, we use the amyloid onset age as our aligning time scale, and what we're observing is individuals that have comorbid Lewy body pathology are showing the cognitive impairment 2 to 9 years earlier than the individuals who doesn't have those comorbid pathologies. So it's a longer story actually, but what I want to show or kind of show some evidence that it's a very complicated landscape. It's very nonlinear, and some of classical statistical analysis approaches are not capturing that nonlinearity and the interactions, the dynamic interaction of some of these biomarkers. It's like someone being amyloid positive doesn't necessarily predict their full progression of the disease for 5 or 10 years. As they progress, different things are introduced, they're exposed to different insults like co-pathologies, or it could be some environmental insults, and that totally changed their progression profiles, and those are not currently captured. And I want to go back to the clinical trial idea. This is a cartoon illustration, but just to give you why this is important from individual perspective. So this is just, let's say you're doing a clinical trial on Alzheimer's disease, and you're saying that your treatment will slow down the decline by 50%. That's what you're testing. And if your participants are clean participants, they only have amyloid and beta pathology in their brain, all of that cognitive decline will be explained by this AD biomarkers, AD pathology. And what you actually will met, like if your treatment is 50% accurate, efficient, then you will actually see that perfectly, and you will see that 50% decline. But in reality, what we're seeing is this profile that varies from subject to subject. That includes, just 10 seconds, just that includes, that could potentially include different co-pathologies, and the contribution of the co-pathologies can vary from subject to subject. And in that case, if your treatment is acting on that AD pathology, it's only going to be 50% efficacious on that portion that is attributed to the AD pathology. not the rest. So in reality, what the trial will be able to detect is rather than showing that 45 to 50% slowing down, that is expected, it can only show the slowing down maybe up to 25 to 30%. And that's exactly what we're seeing at this point with those 3 FDA-approved treatments. They can only show that, and it could easily be the ceiling effect because of these treatments are only targeting the AD pathologies, not doing multi-treatment approaches. And I'm going to disclose one thing. I'm one of the core PIs for Annie. So if there's any questions or challenges with Annie, please find me. I might regret that at the end of this meeting, but I'll be here to answer your questions. To me or?
Yeah, great talk. I think there are several things puzzle me a lot because I know they started to change the AD diagnosis, right? By using ATN type of biomarker.
Not clinical diagnosis, the research definition has changed.
Basically, there's a lot of clinical trials, all the different studies, because many studies already finished. And like ADSP, all these things still use the older diagnosis, right? And if you encourage people to use this one, but there's all the studies already, we don't get all kinds of biomarker information at all.
So I just want to be clear, so diagnosis is still clinical and cognitive. It's not based on biomarkers or the biology. It's just that they need to have like this, they need to have a certain CDR, they need to fall in certain MMSE, they need to show memory impairment. So that's still the clinical diagnosis. What is proposed with the staging, which we just had a new one actually a couple of months ago published, is a research definition. So I agree, like that there's all these previous observational cohort data collected with no PET imaging data or no CSF data, but there are newer cohorts they're collecting. So I just want to be clear, the diagnosis hasn't changed. It's still the same old diagnosis. It's just what kind of biomarkers we're using to biologically define and biologically understand the underlying mechanism that is changing.
I know that the JAX has this kind of documents about this kind of new diagnosis definition. The publication, I think, it posted on the website.
Yes, it's
that's the one that only influence the research.
That's only for research.
Not for the.
It's not used in clinical practice, no. No. Which is a good challenge, actually. So some of my work and some of the studies we did with clinical trials is actually using observations to impute these missing biomarkers. That is very like AI, machine learning, and statistically challenging problem. And we work with a couple trials where we imputed the missing amyloid or the tau information, which allowed them to go back and look at their trials from a different perspective with that biological definition available.
I also have a question for Dr. Tosun. It's a great talk. I'm just curious. So for the biomarkers, you have amyloid, tau, PET imaging, MI. Would the FMI be useful for adding additional information? So what's the research? What's the field think about the FMI?
So I'll give two answers. One practical, one more scientific. So I'll start with the scientific. I think it's important, especially like understanding the connection between biology and the function, right? Again, I'm going to give this example from the current clinical trials. If you, they all show the disease-modifying effect of their treatments. It's removing amyloid, it's slowing down the tau, but it's not showing the expected clinical improvements. There could be many explanations for that. One explanation could be, as I mentioned, the heterogeneity and the contribution of the co-pathologies, but another explanation is the delay, right? There must be something need to happen functionally in the brain before we can see the change in the symptoms and the cognitive decline. And one way to capture that intermediate step is using functional imaging modalities, not relying on subjective cognitive measurements. So from that perspective, I believe that there is a big gap, big need in terms of identifying the right functional measures and proving that they are good enough to monitor the disease progression. From technical perspective, I think it's too challenging because of the challenges with, especially with fMRI imaging. It's very difficult to set up in multi-site setting. And it's also because of the noise and the variability, it's very difficult to track the longitudinal changes. Those are two important things to have as a biomarker in a clinical trial. So if you can address those challenges, John is not agreeing, but
I agree.
yeah,
Yes.
I think it's quite challenging. I don't think fMRI will be the answer.
Question at the back.
Hi, I have a question. I was wondering if you have any comments or suggestions on clinical trial design for early detection of the Alzheimer's disease, because I think these kind of, in terms of like how to proper select the participants and also the biomarkers, because those like amyloid beta and tau protein, they probably will be measured in the mid to late stages, and it's very money consuming, I guess. So do you have any like suggestions and comments?
Yeah, we can chat separately if you have a particular design in your mind or particular cohort. I didn't talk about fluid biomarkers in my talk, and as I mentioned, especially plasma biomarkers, the peripheral plasma protein measurements are the new kid in the block, and but has been extremely promising. Especially some of them are changing quite early. They're saturating really fast, but what we're observing is some of these plasma biomarkers are changing quite early, even before we call someone as amyloid positive on a scanner. So there are definitely some alternatives, and there are actually some studies, like I was just looking at A4 and AHEAD studies actually, because they've been actively using plasma biomarkers to screen for their participants. Those are prevention trials, focusing on cognitively unimpaired individuals at risk for Alzheimer's disease, and they've been successfully using and optimizing some of these plasma biomarkers to identify this high-risk participants without doing PET imaging. But I'd be happy to discuss more.
Thank you so much.
There's two questions. One is for all these ATN type of the biomarkers, you're talking about a large variation account for this clinical decline, like 50% or something like that. This is the measure at the baseline or what? What are these measurements
These are at baseline.
that are measured at the baseline?
Yeah, and I'm looking at like because usually clinical trials are about 2 years. At least the disease modifying treatment trial. So it's looking at baseline biomarkers and how if they're explaining 2-year change.
Yeah, another follow-up question, because recently I saw some papers like JAMA, they talking about the blood biomarkers. Right?
The platform.
And if it seems like they are greater predictors for the disease. In any case, would it be these blood biomarkers will replace most of these like ATN biomarkers, because the clinical is so much cheaper?
Yeah, so that's what I just mentioned, the plasma biomarkers or the blood biomarkers. Some of them are used right now, especially for the prevention trials to identify amyloid positive individuals. I should say they've been cautious. What they're doing is they're doing the initial screening, but they still acquire the amyloid images. But even then, there's a huge reduction on the cost because the number of PET images you need to do is reduced. We are still learning about the blood biomarkers. They are, if you look at the data, the published data, they are good at predicting who's positive versus who's negative for amyloid or tau. It's a binary thing. but it's not, there's still no clear data suggesting if they are actually capturing the disease like the pathology progression properly as a continuous measure. That's one thing we're still working on. The second thing is location is important. And this is again, it comes back from the recent clinical trial results, especially the trial result from the DIAN, that is a dominantly inherited Alzheimer's network study. It's there again, the treatments are able to remove amyloid, but there's huge heterogeneity and some individuals don't show any response to that. And there's again, could be lots of explanations. And one of the things is it might not be the removal of the amyloid, it might be related to where did we remove the amyloid, at what stage did we remove the amyloid. And those kind of information we're still not able to get from the blood or the plasma biomarker. It's that the location and also the stage. They can be amyloid positive, but they could be amyloid positive and then earlier, like their chronicity of the amyloid could be lower, or they could be amyloid positive, but had the amyloid for maybe 10 years, 20 years, and their response going to be different, and we cannot get that information from the plasma yet. So still in practice, we're doing that triaging and then still getting the amyloid PET. Sorry, I'm just,
Okay.
I don't want to.
It's time for any other questions can occur, right? Yeah, right. Thank you.